Okay, it's great to see all of you here and to have the opportunity to talk to you a bit about genetic technologies. I'll try and keep it light today because we'll be talking not only about some of the technologies we currently use in New Zealand, but also a few that are on the horizon that we have. Our industry will have some decisions to make about in the coming months and years. So, along the lines of keeping it light, let's start with a game. This is the trolley problem. So, this is a thought experiment used in psychology to help understand how people differentiate between different ethical problems. So I would like you to play along. The classic example goes like this. A trolley is heading towards 5 people. You can pull a lever to divert it to another track, killing one person instead. What would you do? Who would pull that lever? Show of hands? Okay, again, we see most people in the audience. That's pretty typical of this question. You see about 3/4 of people, wherever this is asked, say that they would pull that lever. And what they're doing is, your reasoning probably went something like this. Either way, we're going to end up with a terrible situation, but by acting, I can achieve the least terrible outcome. And that's what we call utilitarian reasoning. If you decided not to raise your hand and do nothing, then you'd actually have the law on your side. The idea that we can kill one person, even to save others, is actually not the way the law works. It follows something called deontological reasoning. Deon means duty in Greek, and the idea is that you should do the right thing, and the consequences are not really in your hands. So you can't, but you'll see that people tend towards either sometimes they've got to bend the rules to make sure we get the best outcome, and there are other kinds of people who tend to have this kind of, it's really important that we follow the rules, and if in this case it doesn't achieve the best outcome, then at least I did my duty. Okay, let's take a bit more of a personal one. How about this? So in this case, you pull the lever, but you divert the trolley onto a track that you're tied to. Who would pull the lever now? Oh, there's got to be some hands. All right, you see, so it's easy to be utilitarian about other people's lives, but our own personal experiences are so visceral that many people who would take a purely utilitarian approach kind of have these complicating values that about how much my experience matters to me, and it's much harder for us to put ourselves in the perspective of somebody else. Okay, how about this one? So the trolley is heading down towards this rich man. He offers you $500,000 to pull the lever and divert it onto another track. Who would pull the lever? Okay. Or about half people. So we get back to that utilitarian approach, because it does feel like it's utilitarian again, right? That either situation has the same outcome, we lose one life. It's only the money that tips the scales one way or another, and so most people choose that. And Jonathan Haidt illustrates this fantastically in his book, in which he describes our thinking as being like an elephant with a rider, where the elephant, our intuitive mind, is actually making the decisions, and the rider, our reasoning, is kind of going along for the journey and trying to explain the reasoning after the fact. How about this one? The trolley's heading towards 5 people who tied themselves to the track. You can pull a lever and divert it onto one person who tripped and fell on the track. Who would pull the lever now? It's the same as the first one, right? But in this case, we're really testing that all lives are equal hypothesis, aren't we? In this case, if you manage to hold on to your humanitarian scruples for that one, let's try this. 5 very old people, who would pull the lever and divert the trolley, really, onto a track that has a baby lying on it? It's hard, isn't it? I mean, many of us start off with this utilitarian thinking, but then our personal experience, loss aversion, and principles like protecting the innocent kind of complicate the factor, and it becomes really hard to make pure decisions, and as you can see, there's a spread of opinions. Right, keep those ethical questions in mind as we start to think about crop improvement. Today we're facing a trilemma in our industry. This is the world's most planted plant crop, Cavendish banana. It's being absolutely devastated by Fusarium wilt and is expected to go extinct in the next decade or so. Same thing happened with golden kiwi fruit, and this is a critical vulnerability of clonally propagated crops like grapevine. Not only do we have a warming climate, but we're also seeing increased volatility from season to season. And customers are changing their expectations. Young consumers drink less wine, but also they value sustainability credentials and are willing to pay more for them. Last year's Numisma report found that 93% of people born after the year 2000 expect sustainability credentials on wines. So rising disease pressure, unpredictable seasons, and customers demanding fewer sprays, less fossil fuel, and lower CO2 emissions. The typical approach to address these kinds of questions is breeding. And classically, breeding, as it's shown here, this is a photo of the Geisenheim Grape Breeding Institute in Germany. It's a very long and slow process. They have 40 scientists working there. They've been going at it since 1872. They started doing the first powdery mildew-resistant crosses in the 1920s and made the first commercial release in 1996. So it's a very slow process. Just to introduce one new trait is expected to take 25 to 30 years of breeding. And this is why. Say you've got an elite variety like Riesling, and you want to introduce a trait like resistance to powdery mildew. You've got to get a wild relative that has that trait. You then cross it into the elite variety, and all of the offspring, you collect the seed, you plant them out, they have half their genetics from each parent, and half of them will carry the resistance gene. You then take one of the plants with the resistance gene, you have to grow it up to maturity, assess it, check that it's got the right traits, and then you pollinate it again back to the elite variety, and in the subsequent generation, you've now halved the contribution of the genetics from that wild variety. And you keep doing this generation after generation, about 4 years at a time, and after 6 or 7 generations, you end up with something that looks like this. The majority of the genetics are from the elite variety, and you've retained about 2% of the genetics from when you crossed into it. But 2% is still quite a lot. In grapevines, there's more than 20,000 genes, and so 2% of the genetics from that wild variety means that about 400 different genes have just kind of come along for the ride, and it's not really worth continuing to breed to try and get rid of them. Another approach is clonal selection, and that's the biggest piece of work that we're doing at BRI at the moment in our Sauvignon Blanc 2.0 program, and we have a quick video here to give you an update of how that's going.
We're here at Bragato Research Institute's new selection vineyard that houses our 6,000 new Sauvignon Blanc clones that were planted here in December of last year. The aim of this project is to develop new clones of Sauvignon Blanc that are better suited to our changing climate and to problems that the industry might face going forward.
So this trial is a way to genetically diversify the Sauv Blanc we have in the country. It's basically a randomization of the genes that we have, and that looks like 6,000 unique vines in the vineyard here, and yeah, we're trying to find the needle in the haystack.
All of these clones are unique genetic individuals, and we only have a single copy of them, so we're really trying to make sure that they all survive through to maturity. Part of what we are doing in the grapevine improvement team is screening them to determine which ones are of the most potential benefit to the New Zealand wine industry. The plants here, some of them are a year old, so they have spent a winter in a tunnel house, and we're going into their first winter in the ground. The others are about 9 months old, so every plant here is going into its first sort of full period of dormancy outside.
They've just had their foliage wires put up, so they've got lovely canopy to grow up into, they've got frost protection going in. They get lots of love and care. Because it's small, we can afford to really focus on each plant.
Coming into the next growing season, we'll be measuring certain traits. We've already started doing some screens. We've collected leaves from all of the plants and have had the DNA extracted, and we can use sequencing techniques to start to determine which ones might be interesting candidates to look at in greater detail. The key traits that we're interested in are resistance to powdery mildew, water use efficiency, and frost tolerance. We're facing a changing climate, which will affect how we're able to grow our grapes. There'll be increased disease pressure, changes in the availability of water going forward. So this vineyard is really important to help us build resilience in the New Zealand wine industry.
Exciting, isn't it? That's Matt and Amy, two of those, the team of brilliant scientists we have down in Lincoln. But the question now is, are we ready for the next generation of vines? So there's been a lot of change in crop improvement over the last few years, and 3 key technologies are driving that. New genetics, robotics, and AI, kind of closing the loop between selection and breeding. Shortly after I started with BRI, in 2020, Professor Kai Vos-Fels took over leadership of Geisenheim's grape breeding program. And he's been completely revolutionizing the way they do breeding, which has been going, as I said, for over 100 years, to take advantage of some of these new technologies. just a few weeks ago, his team published this summary of new approaches for grapevine breeding that they're focusing on, including things like gene editing and automated selection. So where do these new technologies come from? Well, the classic one people know about is transgenics, or you might not know it by that name, you might know it as GMOs. And the trans part in transgenics means a cross. It means taking genes from one species into another species it couldn't breed with. It was found in nature in a bacteria called Agrobacterium, which transfers one of its genes into a plant, and that gene causes the plant to then produce a substance called nopalene, which the bacteria uses as its foodstuff. And so researchers found that you could take that gene for nopalene production and give the bacteria a different gene, and that bacteria would then put that gene into the plant. So for example, you might take that gene for powdery mildew resistance and put that directly into the plant without going through many generations of breeding. And at the same time, you might take other genes, ones to increase the yield or introduce disease resistance to another trait, and put them all directly into the organism without carrying along all those genes that you were crossing it with. That means it's faster, and it's also possible to breed with genes you don't even have. We don't have any any vines that are resistant to Pierce's disease in this country, but we could get the genes in and directly put them into the plant. And that's been done. So there are grapevines that have been produced with powdery mildew resistance, some with Pierce's disease resistance, and this transgenic approach of putting genes directly into the vines. The other technology you might have heard of recently is gene editing. And this has the unfortunate coincidence of also having the acronym GE. Typically, you would have heard of anti-GE protests from the 80s and 90s. Gene editing wasn't even invented then. GE then stood for genetic engineering, a synonym for GMOs or transgenics. Gene editing is a more recent approach, which again was found in nature. Bacteria have an immune system by which they can recognize viral infection and remove it if they're exposed to the same one again. They basically store a little piece of RNA called a guide RNA, and that binds together with a protein called Cas to digest the viral genome when it's infected. And so again, scientists thought we could take that guide RNA and we could swap it out with a piece of RNA that targets any other gene. And in doing that, so you can drive that Cas enzyme, if you put it into a plant or another organism, to make a targeted mutation, a break in the DNA in any other species of interest. And so I've been thinking about how to make this more visual for people who aren't familiar with genetic code. So instead I've swapped out the genetic sequence for an alphabet sequence, something we're a bit more familiar with. And let's imagine this first vine has a genetic code that says this vine is not disease resistant. Gene editing might look something like this. A chemical change to just one position in that genetic sequence makes the trait completely different. That T has been changed to a W, not because a W was introduced, but because a chemical change at one location has altered it. Another type of gene editing might be this, to introduce a deletion. In this case, the NOT gene has been deleted. Again, nothing has been introduced, but the removal of that gene completely changes the traits that that plant has. Another one that I wanted to draw your attention to is called cisgenesis, and this is when you take a gene from something that the organism could breed with, but rather than going through many generations of crossing, you just take the gene of interest and put it into the host organism that you're looking for. So it's a way of sort of accelerating the process, even though the outcomes in these cases are the same as what you could achieve through traditional breeding. So gene editing has been done in grapevines. It's been used to make grapevines that are resistant to leaf roll virus, grapevines that are resistant to powdery and downy mildew, and both in Europe and the US, there are field trials for these gene-edited vines already underway. The key reason that there's been a lot of uptake for this technology is that it's very rapid, it happens one generation, it's precise, and it's very adaptable. By swapping out the code of that guide RNA, you can target different genes for mutations. Mutations are something that happens a lot in nature. DNA is damaged when cells divide or when it's exposed to environmental stresses, and it has to be repaired, and sometimes that happens incorrectly. Or just copying the DNA when a cell has to be replicated can make mistakes. And so if you took any two vines from the vineyard and sequenced their genomes, you would find mutations, tens, maybe hundreds of mutations between those vines. These technologies make the same types of mutations, but in specific places. And that means there's no real markers to detect. In the case of transgenics, you've introduced a foreign gene, so you can sequence the DNA and say, hey, there's a gene here that's not supposed to be in grapevine. But if you sequence the DNA of a gene-edited vine, you just find lots of mutations. And there's no way of saying which one was intentional, which ones were just random. The new clones produced in this way are also, the new plants are new clones. They haven't been crossed out with some of the variety, so they don't need the varietal rebranding. And because they don't introduce any foreign genetics, or when they don't introduce any foreign genetics, they're often not regulated as GMOs. And so in many of the countries we trade with, they're not considered GMOs. And in fact, the Food Safety Authority of Australia and New Zealand has proposed that if there's no foreign DNA, they wouldn't be regulated as GMOs when it comes to labeling. So foods could be sold in New Zealand and not labeled as GMOs, even though if we were to produce them here, that would be considered GMO. So for this reason, our government has proposed a new regulation that kind of catches us up with where our trading partners are. And you would have heard about the Gene Technologies Bill. It's gone through its first reading in Parliament and Health Select Committee, which New Zealand Wine Growers made a submission to. And the idea isn't actually to decide which technologies are allowed or prohibited at this stage. Instead, it establishes a risk framework, 4 tiers of risks, and subsequently, it'll then fall to a regulator to decide which technologies and which outcomes fall in the different risk tiers. So the first risk tier is technologies that are exempt. If the outcome of the technology produces the same as what could be achieved through traditional breeding, then that wouldn't be regulated. There are then 2 tiers of low risk or very low risk outcomes that would be regulated, but would either be require or not require notification. And then for anything that involves transgenics or clinical trials or field trials or commercial releases, these would still need to be licensed, which requires applying for the license and opportunity for public submissions. There are a lot of concerns people have with changing legislation around gene technologies, and we unfortunately don't have time to discuss a lot of them here today. Some of them are around safety and the precision of the technique, and with genetic sequencing, we can now actually have a look at every single genetic change that's happened in a gene-edited crop. Often people are concerned about the ecological effects, like will there be cross-pollination if my neighbor chooses to plant gene-edited plants? For our industry, that's less of a concern because we don't grow grapes from seed. So it would be analogous to thinking, I plant Sauvignon Blanc, my neighbor's planted Pinot Noir, am I going to get Pinot Noir in my wine? No, you're not. The genetics that comes in, even if it pollinates your plants, that ends up only in the seed, and we don't grow the grapevines from seed. That doesn't contribute to the skins or the berries or the juice. Others are around trade, market acceptance, and the bill is designed to align us with our trading partners, but what about consumer acceptance? And this is a bit more of a difficult one to address because consumers don't have such a binary approach to gene editing. The sort of anti-GE protests have changed to more of a subtle focus on sustainability credentials. And almost all of these gene editing techniques that have been done in grapevine have been done to increase sustainability credentials, like I say, to reduce the use of sprays. And so what we have here is a much more complex question that we all need to discuss. And so, for example, if you interview New Zealanders about what they think about new genetic technologies, you find they're almost evenly split into 3 groups: people who oppose, people who support, and people who are not very sure. But if you instead ask them who supports the use of gene technologies for controlling pests and diseases, or increasing food nutrition, or decreasing the cost of food production, what you actually find is most people are supportive of these technologies. And so what we have here is not a question of gene editing or not gene editing. Instead, we have a trolley problem. There are 2 paths, and as a community, we need to decide which path we want to be on. Do we want to be on the path that has reduced spray use but accepts gene editing, or do we want to stick with what we're already at? And to illustrate where we're really at with this trolley problem, here's one final one for you to consider. Oh no, a trolley is heading down towards 5 people. You can pull the lever to divert it to the other track, killing 1 person instead. At least that's what you think is happening. You forgot your glasses and you can't see well. What do? Who would pull the lever now? It's a much harder decision, isn't it? And whenever this is asked, it's almost split 50-50. And this really illustrates how our willingness to act to achieve the best outcome is limited by our knowledge and our understanding. So wherever you stand on gene technologies, whether you feel like I take a utilitarian approach or whether you acknowledge that your intuition is actually driven by a much more complex set of values and conflicting priorities, I hope we can all work together to close the knowledge gap and hopefully make some better decisions. Thank you very much.